conjugating the carrier protein to them. It changes their properties. They now become T-cell dependent antigens, showing affinity maturation, memory, class switching, and becoming immunogenic in infants. This is just as an example of the remarkable success of these vaccines. People, I think, know Prevnar very well, but in perhaps less well known is the effect of the meningitis vaccine. Sub-Sahara Africa has a meningitis belt where they get periodic waves of meningitis, shown here over the last 50, 60 years. And you can see with the arrow, with the introduction of the conjugate vaccine Menafric made by the Serum Institute of India, essentially this killed the epidemic waves. It's a highly efficacious vaccine. And this was actually a partnership between the NIH, the FDA, and the Serum Institute of India, with funding from PATH and the Gates Foundation. It's a remarkable public-private partnership with some remarkable results. My area, as I mentioned in the beginning, is in conjugation chemistry. And I'm most known for the development of the CDAP chemistry, which is a cyanylating reagent. And it is used in several commercial vaccines, including ones from GSK, Pfizer, and the Serum Institute of India. A number of other places have it in clinical, are using it in vaccines that are in clinical development. Notably, the Serum Institute of India's Pneumosil vaccine is the lowest cost pneumococcal conjugate vaccine, priced for Gavi priced at $2 a dose. The Pfizer and GSK pneumo vaccines are currently a little over $3 a dose. It's now WHO pre-qualified. Sorry about that. And it is being sold now across the developing world. In phase 3, is there a new generation meningitis vaccine with 5 serotypes, A, C, Y, W, and X. It uses chemistry that I developed and was largely developed to circumvent some blocking IP. And my understanding is from the preliminary data that this vaccine is looking very good. It's expected to be introduced in 2024. So another class of conjugate vaccines beyond the polysaccharide ones are haptens, small antigens that are poorly immunogenic. These can be chemical, peptide, oligosaccharide, by chemically conjugating these to the carrier protein, they provides the T and B cell epitopes to create the immunogen. Now, there are only a limited number of carrier proteins that are commonly used, generally BSA or KLH at the research level, but these are really not suitable for clinical use, and the KLH as a natural product is difficult to commercialize. Although there have been a number of phase 1 and maybe some phase 2 vaccines that do use KLH. In licensed conjugate vaccines, there's really principally 2 carrier proteins, tetanus toxoid and diphtheria toxoid. But these are actually fairly difficult to obtain for vaccine use in the quantities that are needed. And for experimental phase 1 work, they're quite expensive as well. On top of that, these are non-homogeneous products, particularly tetanus toxoid, as I'll discuss later, and that causes problems for the synthesis of conjugate vaccines. The other commonly used carrier protein is CRM197, a genetically detoxified diphtheria toxin. This protein is 58 kilodaltons, has a number of different features to it, including binding to the EGF receptor, EGF receptor, And it has actually been used in cancer treatment or evaluated in cancer based on that binding function. Notably, it's used in Pfizer's Prevnar vaccine. It has a single mutation, which renders it more or less enzymatically inactive. But unlike toxoids, which are treated with formalin, this protein has its full complement of lysines, of which 20 are accessible and 7 are most commonly exposed and used in the conjugation processes. There are also 2 disulfide bonds, and the protein is protease sensitive. It nicks, and this is part of its activation process of binding to its receptor, transposing across the membrane, and then entering into lysosomes and turning itself inside out. And one result of it turning itself inside out at low pH, it occurs at low pH, and it in solution, at least when it does this, it will aggregate and denature. There are a few CRM sources. The natural version is secreted protein from Corynebacter, which is a slow-growing bacteria, and generally, with some exceptions, most groups end up with 50 to 100 mg per liter, relatively low. About 15 years ago, Phoenix, in conjunction with the Serum Institute of India, developed the first recombinant CRM in their Pseudomonas florensis strain, and this is used in their pneumocil conjugate vaccine by the Serum Institute. It's the best known recombinant one. They've been around the longest, and it is also licensed to Merck and part of their Merck's 15-valent pneumococcal conjugate vaccine. It has been priced fairly high and not suitable at that price for use in vaccines for low-income countries. There are some other groups that have been expressing CRM in E. coli, principally in the periplasm, and some in inclusion bodies. About 7 years ago, Fina Bio began working with PATH to develop a new E. coli expressed CRM197, which we did succeed at, and I'll discuss that in a moment. We market it under the name EcoCRM for E. coli CRM. The key to success to this was a engineered BL21 E. coli that has an oxidative intracellular environment. There are a few other strains that have this intracellular environment, notably the origami and shuffle strain from NEB. These are crippled strains and don't really grow to very high levels. What's remarkable about this, our BL21 with the oxidative environment, is that it grows to very high densities, 200 to 300 grams of protein per liter. And this makes it commercially viable to express recombinant proteins. So our CRM with its disulfide bonds is expressed as an intracellular properly folded soluble protein, and we make it at about 2 grams per liter. According to the Gates Foundation, at this level, we've essentially removed the cost of the CRM as a cost driver for pneumococcal conjugate vaccines. We have done extensive biophysical studies of our EcoCRM. Some of this work was done with Redshift Bio, who have a FTIR instrument that called that works on a microfluidic scale. And they have done the higher order structure comparison with the established Phoenix CRM, and shown that EcoCRM has the identical structure. And this the manuscript for this is in press in Pharmaceutical Outsourcing Journal, and that should be out actually any week now. A much larger and more comprehensive study was undertaken with funding from PATH at David Volkin's lab at University of Kansas. They compared 5 different CRM197s from different expression systems and different manufacturers. And I will just give you a taste of what they found. But the blue and the brown or black marks here are the Fentanyl and the EcoCRM respectively. Those were available in GMP or at least GLP levels. Interestingly, the native creme, which is the orange one and the one that looks a little bit different here, was somewhat actually misfolded. But it was the least. It was made for research use only. So this higher order structure, circular dichroism, the UV intrinsic and extrinsic fluorescence showed that the protein was properly folded up. There is a lot more detail showing the correct disulfide bond forming, a lot more thermal stability data in the Hickey manuscript. We now have clinical grade EcoCRM available. This just shows some of the general quality attributes of it. We have now done six manufacturing runs, with a 7th going on this week. This shows that the data more than matched our expectations for the product. Notably, here it has very low endotoxin levels for the conjugate vaccine. What's interesting is we're taking whole E. coli lysate, and in 2 column steps, we're reducing it to this level. There are a number of vaccines in clinical or preclinical development, covering a wide variety of areas: malaria, cancer, COVID, Group B strep, strep pneumonia, and vaccines for drugs of abuse. All of these there are many others that are in still at the research level, but all of these are at the stage of being transferred to a CMO for clinical manufacture and clinical trials. The one that's furthest along is one for COVID. I think in my contract to speak here, I was obliged to mention COVID at least once. This is it. So there are 2 conjugate vaccines that who our clients are using for COVID vaccines, and hopefully they'll be successful. So I'm going to cover now an example of a chemical hapten conjugate vaccine that uses EcoCRM. The concept behind a vaccine for substance use disorder is that you induce antibodies against the drug. You have circulating antibodies, and then these antibodies will bind the drug and prevent it from reaching the brain. There was a lot of early work done with KLH being something that you could buy GMP, and so there was interest in that, as well as tetanus toxoid, working with Marco Provatoni at University of Minnesota. He made conjugates with our EcoCRM. It was very nice. Unlike the KLH and TT, he could do mass spec on them and actually determine and characterize the conjugates made. And he found that compared with KLH here, the CRM conjugate was further able to block the uptake of oxycodone into the brain. Another vaccine for drugs of abuse is one we're doing with University of Houston and Baylor College for a fentanyl CRM conjugate vaccine. And for this experiment, rats were immunized with either saline or with the fentanyl CRM conjugate, and then subsequently exposed to fentanyl. The control rats were stiff as a board. This curved tail is apparently characteristic of these, but basically in a catatonic coma. They showed they were comatose and fairly immune in the tail plate, tail flick hot plate assay. In contrast, the vaccinated rats were happy and cheerful, doing their rat things, normal behavior, and sensitive to in the tail flick assay. I do have a brief movie here of you can see the comatose rat at the right and the happy little rat on top. So these vaccines are proceeding. We are working with 3 different groups doing vaccines for substance disorder. We're very happy to be part of this important area. So another area that we are working with in conjugate vaccines to promote conjugate vaccines is that we have taken our CRM197 and pre-derivatized it with cross-linking reagents to facilitate the research in vaccines, because this makes it very easy to couple haptens to it. It's very easy to use mass spec to determine the peptide loading. We use that both for ourselves and have this commercialized. And our intent is to prepare CRM maleimide as a GMP product so that vaccine developers won't have to spend all the time and money to develop that product on their own. As an example of a peptide conjugate made using one of our CRM maleimides products, cattle tick fever disease. This is a little bugger that causes cattle tick fever. It's a big problem along the border. You can see how nicely we're able to characterize our conjugate. And cattle have been immunized with these conjugates. They did have 6 in one group, but that was a bum steer. The peptide EcoCRM has induced a very nice immune response. We also compared a tetanus fragment, which also induced a good response. These cattle are going to be challenged in the next couple of weeks, and hopefully the next time I talk, I'll have some results from that. So again, this makes it, having this CRM maleimide ready to go makes this kind of vaccine development extremely easy. I mentioned before the other carrier protein, tetanus toxoid, that's widely used. Tetanus, although people think it is generally, it's available, who cares? It's a cheap vaccine, it's been around forever. But the fact is that there are actually only a limited number of large-scale manufacturers. The quality, every manufacturer has their own process. It's a fairly junky protein. As part of our contract work with vaccine companies around the world, I was able to obtain tetanus toxoid from a number of different manufacturers and different lots. These are size exclusion chromatograms, and you can see that the percentage of tetanus in most of these, of monomeric tetanus, ranged from that middle one, C, is a veterinary vaccine that was less than 10%, up to about 60%. You can see that these solutions are brown and junky, showing a lot of impurities. There's a more complete analysis of this that you can download on a poster from our website. We began working with the Medical College of Wisconsin on commercializing a genetically detoxified tetanus toxin with 8 mutations. This work has been published by Dr. Barbieri, and I'm not going to pronounce the other name, but this has 8 mutations in each of the 3 major receptor areas. It's more complicated than CRM, which has a single mutation, but because tetanus is so incredibly toxic, that they needed to make many mutations in order to reduce it. It's about 1/50 millionth as toxic as the native protein. We used our same expression system in same oxidative environment E. coli, and express the CRM. We made the 8-MTT. We have so far made it at over 0.5 gram per liter. This is a protein is 3 times larger than the CRM. It has 5 cysteine pairs. So I again find it fairly remarkable that we're able to make this much protein. We have one small trial that I can talk about now. We have several that are ongoing, but so far it looks like it is as good or better than the native tetanus or the regular tetanus toxoid as a carrier protein. So the last short segment, I'd like to just discuss a little bit about our business model and my personal mission. I've been working in the field of conjugate vaccines for my entire career. And I set out when I founded my company to focus on promoting the development not only of conjugate vaccines, but affordable conjugate vaccines, such as we've done with the Serum Institute of India. Our business model is no outside investors. This means that we don't need to make money for other people. We just need to make enough money for us. This allows us to do licensing and agreements that would not make sense for investors who want to make money. And it has been challenging. And in the last slide, I'm just going to take you through a little cartoon that shows how we were able to make GMP CRM without having any outside investor money. So we are entirely self-funded. As a result, we have very little excess money. We partnered with PATH, as I mentioned, to create CRM. They gave us some funding. In turn, we gave PATH a royalty-free license for use in Gavi countries. In turn, we subsequently licensed to a company out in Seattle, InventPrize, that also is developing affordable conjugate vaccines. With PATH, we provided a contract to InventPrize to scale up our EcoCRM production. The Gates Foundation is now funding InventPrize for their pneumo and Group B strep vaccines, conjugate vaccines for Gavi countries. As a result, they needed the CRM that we provided them as the carrier protein. And out of that popped the GMP EcoCRM, which then went back to Fina BioSolutions, which we can sell and then generate the money that we need to sustain our company. And that is the not for much profit business model. With that, I'd like to wrap it up. Thank you for your attention and tell you that we would be happy to work with you. Thank you very much.
Thank you, Mr. Andrew. So I would request if any other participants or the attendees have any queries, so we can take up.
Hello.
Yes.
Hello, great talk, Andrew. I have a naive question, perhaps. Can you hear me?
Yes.
Yeah. So when you conjugate this haptane to the carrier proteins, right? It can non-specifically attach to any amino acid possible, right?
I'm sorry, say that again.
When you attach this haptane to the carrier proteins, right?
Mm-hmm.
So it can non-specifically attach to many other potential sites, right? Nucleophilic sites. So it doesn't matter where it attaches.
It's random.
It's random.
So there are, as I showed there, it's random coupling depending on the chemistry.
Right. So do you use the random mixtures then?
Yes.
Okay.
So there are really
them, right?
Yes, there are 7 most accessible lysines.
Right.
People who have studied, broke down used mass spec to study. Right. I think this came out of GSK Siena.
There could be a number of haptanes could be a couple, right? A number of haptanes could be coupled to the protein, right?
You typically end up with between 5 and 15 if you're using the lysines.
From the mass spec, you get that information, I guess.
Yes.
Right? Okay.
Yeah.
Thank you very much. Nice talk.
Thank you very much.
Do we have anybody else? Any queries? Please raise your hand so we'll call you one by one. Okay, we have one attendee here who wants to speak. So I request the technical team to just unmute Francisco Micoli in the attendees list so they can speak. Yeah. Francisco Micoli, you're unmuted. You can ask your query.
Can you hear me?
Yes.
Yes, we can hear you.
Okay, thanks. So, Andrew, very nice overview of your work. Thanks a lot. I was curious to know if you have compared the immunogenicity of glycoconjugate produced with the recombinant, let's say, EcoCRM or recombinant TT.
Do you mean, have we done it in vivo to do the
In vivo,
conjugation?
in vivo, I mean, yeah.
No, we don't do any in vivo. That involves
Okay.
a whole nother set of, bringing in the genes into the cell and so forth.
Okay, thank you very much.
Mostly we provide our CRM to groups that are doing that, and they need a standard to compare against.
Thanks.
Hi, Andrew. Can I ask a question?
Yeah, sure. Go ahead.
Andrew, it's Dr. Ritu Chakravarti from University of Toledo. Very good talk. I'm very new to the field of vaccines. So my question to you is, do you share your reagents for the research and development purposes? So if I'm developing a vaccine, can you provide me the hardware that you recommend?
The answer is yes. In fact, we just put out with the Canadian Glyco Group. Are you familiar with the GlycoNet?
I've heard about it, but I'm not part of it. So
we've collaborated with several scientists in that, and in the latest issue of their newsletter, we made the offer to provide free CRM samples to Canadian GlycoNet researchers. But really we'll make it broader. Part of our, we don't make our money really selling the little milligram amounts of, the research amounts. Our primary interest is in developing collaborations with other researchers. Because we don't have our own antigens in general. We go out and collaborate with other people who do have problems, and then we share our expertise in conjugation and in vaccines in general, and partner. In fact, everyone that I discussed here came out of reaching out to looking for collaborators.
So if I'm located in US, so if I want to try this, how should I be going about?
Just go to our website, and any email on there goes to me.
Thank you.
So, but I can extend this to anyone if they are interested, and we can provide them with CRM, for research use.